Je vais commencer en français, déjà pour savoir qui a des gros problèmes de communication en anglais. Il faut me le dire parce que quand il y en a pas. I start in French and then I will switch to English, of course. Parce que si vous avez des soucis en anglais, c'est pas un problème, mais du coup, il faut que je les identifie pour vous faire un peu la traduction de tout ce qui va être dit. C'est compliqué pour moi de faire un truc en temps réel, mais l'idée c'est ça. Est-ce qu'il y en a qui vraiment sont... Voilà. On essaye, et si vous voyez que c'est trop difficile, voilà. Par contre, ça ne doit pas vous inhiber pour prendre la parole. En anglais ou en français, je traduirai. On est là pour avancer dans des thématiques, donc du coup, on va essayer de faire ça comme ça. OK? Est-ce que... je switch en anglais. Second question, so can we just identify who is from care here? Any people from care? Okay, 1, 2, 3, 4. And if I well understood, I have 1, 2, 3, 4, 5, 6, 7. 7 plus 4, it's 11. So according to me, someone is missing. Okay, and because you have something like 15 or 16, no?
Yes.
No? So we can wait a few minutes. Then with from the other master, so from BSG. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9. Here again, some are missing. Do you know where are they? No? Okay. And the last one should be from Cecil, that's it? And here you should be something like 7?
Yes. Yes.
And you are? Hand up. 1, 2, 3, 4. You are 4. So here again, do we have to wait some people that you know they are coming? Okay. Okay. So it's very important for you to assist to this kind of workshop. Why? Because some ask me about the evaluation context and what is exactly the exam. So exam is quite simple since what you will have is some question about the different workshops we are going to do together. So if I well understood and if I remember, we will have 4 different workshops, right? The one of today the one of today is dedicated to telomere, if I will remember, I think. Then we'll have something about inflammation, about senescence, not maybe in the good order, but and the last one will be dedicated to metabolism and oxidative stress and mitochondrial etc. Okay, so all this workshop will consist in discussing around the use and the identification of aging biomarkers, right? And the aim of this kind of workshop is to give you, of course, the opportunity to discuss, to present some things, and to ask some questions. So it's not only just to assist to, today we have something like 2 presentations, the global aim, and I would say that maybe in my, I don't know if I can say that, but you know, in my vision I don't have from you is also the participation we'll do, participation will do in the context of questions. So some people are going to present, I don't know, by 2, by 3 people, and then of course I hope that you will have a discussion, because without discussion that means I can ask you a question regarding the presentation, and if you didn't understand or if you don't want to open the discussion, that means that the question will have no answer, so it could be difficult for you. Okay, so I count on you to ask some questions. Can we have a kind of feedback from people that are online? Just maybe Janet, if you can say if we heard us, right?
Yeah.
Yeah, this is okay for you. Do you see any people? You cannot see me. The camera is right there, but it's going to change when you're going to present. For those that are presenting online, I will ask you to activate your camera, of course, when you present, when you are not presenting. That's quite important to see you and to know that you are ready to work, okay? So today we're going to talk about telomere. So how do we work? We are going to identify the 3 groups. I hope that you get the time, of course, maybe to read at least the abstract of each publication for those that are not going to present. They are going to present, so it's going to last, I don't know, something like 10, 20 minutes, depending about the question, the discussion we have, 30 minutes per group. And between the group, we are going to ask some questions. So that way I don't start by a presentation. And we are going to try to make a kind of presentation, but at least at the end of the day, to give some insight about the topic of today. So today is telomere. I hope that everyone in the room know what is telomere. You know what it is? Okay. We are going to ask the question differently as usual, because usually we ask the question to know what is telomere, or they can be involved in aging process, etc. Today, the question is quite different, since we would like to know how we can measure telomere, what it is exactly regarding the aging phenomenon, and can we consider them as a measure of telomere and associated telomeres and enzymes that are dedicated to build this kind of DNA sequence as a good biomarker of aging. You should know that it's quite difficult to identify aging biomarkers. And in this context, we try to maybe open our mind on different stuff that are directly linked to aging, okay? So today we're gonna ask the question, can we use telomere as a good marker of aging? Which kind of aging, of course? Are there different agings that are not linked to telomere shortening? You know what it is, telomere shortening? How we measure telomere? Can we measure it clinically? Can we measure in the clinical context? And is it something we can do in the team? Of course, this is quite important if you want to translate your research, your basic research to clinical assay. Okay, so it's quite important. Who is going to present today? One group, two group, I guess, and three group. Or you are together?
We're presenting online.
Okay, so we are our group that is online. So today there is no order in the way to present, so we can start by presenting if you want online, if you are ready, and Janine, if your colleague are online also. I don't know if you are all online that you are going to present the paper, first paper.
Yeah, we're both online. Bella, we can go first if you want. Oh,
yeah,
she's here. Okay, sounds
who
good.
is with you? Who is with you in your group?
Isabella De Silva.
Oh, you all need to. You all need to. Okay, you all need to. Okay, good. So what we can do, Isabella, I don't know if your microphone is on, but we are hearing you, but very, very, very weak. Can you speak again?
Can you hear me?
Yeah, that's great. Okay, good. So if you can share, we're going to start by you. And then, of course, all of you are going to take some notes during the presentation. And in case of an understanding, you can interrupt them. The aim of today is to discuss, okay? So you have to discuss with your colleague. I'm here just to try to animate the debate, the discussion, to enrich the discussion and to make it something that we can, I don't know, maybe resume at the end of the day. Okay, so we're going to start by the TF2 paper, and then we will talk about with the two last ones about how to measure the telomere. So Janine, the floor is yours, Isabella. Of course, don't hesitate. If I want to interrupt you, or if your colleague want to interrupt you, I will reach the the yellow hand. Okay. That's right. Before you start, just to know if some of you have some question or not. You have question? It's okay. For those that were late, are you okay with the English spoken version or not? It's okay for you, more or less. I don't want you to be limited by the language. If it's the case, you have to switch in French. Okay, in French. Sorry. If it's the case, really, believe me. Okay. You switch in French because all these sessions are normally made for different masters, and at the beginning, at the beginning, it was not supposed to be only in English. So we switch only in English for care people, of course, but we can also. I think for you guys that coming from VEG or CCS, it's quite good. But this is not mandatory to speak in English because it's not what I done on the syllabus, okay? So don't be stressed by English language, okay? Here we go, Janine.
Yes, can you see my screen?
Yeah, it's perfect. You can
Okay.
go on.
Okay, perfect. So, good afternoon, everyone. Today, Bella and I will be presenting the paper that we were assigned, which is Induced TRF2 Deletion Leads to Aging Vascular Phenotype in Mice Associated with Arterial Telomere Uncapping, Senescence Signaling, and Oxidative Stress. So, to give you a background, the importance of vascular aging and in cardiovascular disease, aging is the primary risk factor for cardiovascular disease. The age-related changes in large elastic and resistance arteries reduce blood flow and increase blood pressure. So, vascular aging impairs nitric oxide availability, affecting the blood vessel relaxation. The accumulation of senescent and reactive oxygen species are linked to vascular dysfunction. So how does this tie in with telomere uncapping and oxidative stress as well as cellular senescence in vascular dysfunction? So telomere caps chromosome ends, protecting them from DNA damage. The loss of telomere protection, which is called uncapping, activates cellular senescence, triggering inflammatory responses. And ROS, which is often released by senescent cells, reduces, increasing vascular stiffness and the risk of hypertension. So TRF2 protein stabilizes telomeres and the deletion leads to uncapping and mimics vascular aging. Therefore, this study used a mouse model to induce TRF2 deletion and observe aging-like vascular changes. So the hypothesis and objectives. The hypothesis was that deleting the TRF2 in mice will cause telomere uncapping and initiate aging-like vascular dysfunction. The expected outcomes included increased oxidative stress, impaired vasodilation, and blood pressure changes. The primary aim of this paper was to determine if TRF2 deletion induces telomere uncapping and will mimic human vascular aging. They also wanted to do a relationship exploration, and this would be to investigate the role of oxidative stress and senescent signaling in vascular aging, as well as to use the module to establish a causal link between telomere dysfunction and age-related vascular changes. So from the results, we find that TRF2 deletion resulted in a significant 85% knockdown of TRF2 mRNA in the arteries. We also have uncapping, which increased by 55% without affecting telomere length. This suggests that structure, not size, matters. So the significance of this result would be that the uncapped telomeres activate DNA damage response, leading to cellular senescence, and this type of structural instability, rather than shortening, mimics age-related telomere damage in the human vascular culture.
Okay, so next they found that TRF2 deletion leads to arterial P21 induced senescence signaling, and the TRF2 negative mice showed a 320% increase in P21 positive cells as well as P21 mRNA expression. They also investigated the P19 RAF expression, and there was no significant change, which indicates that the senescence is not due to mitogenic stress. And the reason they actually investigated the PR19 was to see if there was an influence of P53 activation and subsequent P21 regulation, which may have occurred independently of TRF2. However, this was not the case, so we know that what we're observing is actually due to TRF2 deletion. So next, they found that TRF2 deletion leads to impaired endothelium-dependent vasodilatory capacity in both carotid and mesenteric arteries. And they found that there was a reduction in vasodilation capacity in the carotid arteries by 31% and the mesenteric arteries by 22%. And this impaired dilation was primarily due to lower nitric oxide availability, which was confirmed by a nitric oxide synthase inhibitor experiment. And there was also a decreased microvascular perfusion by 11% and a reduction in glycolytic thickness by 63%. So then here in the first figure, we can see that endothelium-dependent dilation in response to the endothelium-specific vasodilator, acetylcholine, was reduced in isolated carotid and mesenteric arteries from the TRF2 negative mice compared to those from the TRF2 positive mice. And next, we looked at the NO-mediated vasodilation, which was 24% lower in carotid arteries and 26% lower in mesenteric arteries from the TRF2 negative mice compared to those of the TRF2 positive mice. And finally, they looked at the carotid or mesenteric artery dilation to the endothelium-independent vasodilator, sodium nitroprusside, and it was found that there was no difference between the TRF negative and TRF positive mice. And this shows us that TRF2 deletion leads to impaired endothelium-dependent vasodilatory capacity in carotid and mesenteric arteries. So next, they looked at the effect of the TRF2 deletion on in vivo vascular formation. And firstly, what they see is that the microvascular perfusion, which was assessed by measuring the red blood cell fraction across a range of mesenteric microvesicle diameters, was reduced by 11% in TRF2 negative mice compared to TRF positive mice. Next, they went and they looked at the mesenteric microvascular endothelial glycolax dysfunction, and this was assessed by measuring the depth of the red blood cell penetration into the endothelial glycolax across a range of different mesenteric microvesicles as well. And there was actually a 20% increase in the perfused boundary region of the TRF negative mice compared with the TRF positive mice. And this actually signifies endothelial glycolax deterioration of the TRF2 negative mice. And lastly, they assessed the glycolax thickness, and there was a 63% reduction in endothelial glycolax thickness in the mesenteric microvesicles of the TRF2 negative mice. And all of these results together show us that the TRF2 deletion actually leads to an impaired mesenteric microvascular perfusion and endothelial glycolax deterioration. So next, they found that the TRF2 deletion leads to a pro-oxidative arterial phenotype and reactive oxygen species mediated endothelial dysfunction in carotid arteries. There was a significant upregulation of NADPH oxidase and superoxide levels around a 210% increase. And reaction oxygen species were partially dependent on NADPH oxidase activity, which is shown as NOx inhibitors. And there was also a reduced nitric oxide bioavailability due to the superoxide mediated degradation. So next, they showed a 28 and 186% increase in NOx2 and NOx4 mRNA expression respectively in the arterial tissue from the TRF negative mice compared with the TRF positive mice. Likewise, when we look at the mRNA expression of antioxidant enzymes such as SOD1, SOD2, and SOD3 in the arterial tissue from the TRF negative mice versus the TRF positive mice, there was actually not a significant difference between the two. And this tells us that the TRF2 deletion leads to a pro-oxidative arterial phenotype. So finally, in conclusion, these findings support the initial hypothesis that TRF2 deletion-induced telomere uncapping models aging-related vascular dysfunction, and this also confirms the role of oxidative stress in senescence and endothelial impairment. And this also provides us with insights on human vascular aging. It suggests that telomere uncapping is a novel aspect of aging-related blood pressure and vascular changes, and it highlights the potential for TRF2 deletion as a tool for vascular aging studies. So with some perspectives from this paper, there's a biomarker potential. So exploring telomere uncapping as a biomarker for early vascular signs of aging in human. There is a perspective for pharmacological and behavioral interventions, so to target oxidative stress pathways for telomere uncapping, which could slow or reverse vascular aging. However, there is a need for exploring interventions that enhance nitric oxide bioavailability to counter oxidative stress effects. And TRF2 deletion causes telomere uncapping, triggering oxidative stress and vascular dysfunction. And the other key point from the paper is that the changes observed in nitric oxide mediated vasodilation, glycolax, and blood pressure mimic aging. And the relevance of this model is that the TRF2 deletion is a promising model for studying vascular changes with age, with a potential for therapeutic insights.
Thank you very much. Thank you, Janine. Thank you. Isabel. Thank you very much. You did great. I think you did great because I understood everything and it was quite clear. So first of all, before asking, of course, my question, I would like to hear about the question raised by the audience. So we can start the discussion. Let me know if you hear well, and we will have the discussion. So what do you think about this paper? What kind of question do you have in French or in English, of course? Don't forget that the discussion is something that you have to do to try to fix, the part that are not very clear for you. Yeah?
I didn't get why the pro-oxidative phenotype was only found in carotid arteries and not in the mesentery one.
Good question. It was mine also. Do you hear her? Yeah? Can you answer, Janine or Isabella? Why this pro-oxidative phenotype was only found in some arteries? What is your explanation? What is your point of view?
I can't hear. Can you say it again?
Can you repeat me because you are near the microphone? Can you repeat it?
I didn't get why the pro-oxidative phenotype was found only in carotid arteries and not in the mesentery one.
What is the explanation raised by the author? Why there is some discrepancies between different vessels in terms of consequences of TRF2 deletion?
From what I understood, it was because both the carotid and mesenteric arteries would allow for comparison, but the carotid arteries, because of their function and supplying blood and the blood pressure, it would be more important for the vascular dysfunction due to aging. So I think from what they said in the paper was that they focus more on carotid because of the significance rather than mesenteric.
Okay. Do you think that it is an extra question? Do you think that, do you know if the carotid are more, how can I say that, more triggered by senescence and by telomere shortening than other vessels because of their activity or because of their, is it something that you saw somewhere?
No, but from what I understood, because of the background, I understood that carotid arteries are classified as the elastic arteries. So then it would make more sense that they would study it more because that was the main reason they were looking at vascular aging, and they showed that the elasticity results in the blood pressure and the aging phenotype, because that was like one of the first things that I wouldn't say it impressed me, but was stood out for me from the paper was the opening statement that aging is the primary risk factor for cardiovascular disease. So I think that's why I would say like they would look at carotids because of that elastic link as well.
Mm-hmm. But this is just to enrich the discussion. It is well known that there is how can I say that, a high occurrence of, first of all, of senescence, which should be linked also to telomere shortening in endothelial cells. In my hand and in other hand, also in other paper, we can easily see that even in young individuals, you can find, firstly, telomere shortening and senescence, associated senescence in endothelial cells, whereas you cannot identify telomere shortening in other cells. So it seems that these are the first cells to become senescent and to be observed in terms of telomere shortening compared to other cells. We don't know exactly why. Some argue that it could be also because there is a kind of large turnover of these cells. They are also highly solicited, but we don't know exactly why. But for instance, if you are working on metabolic pathology such as obesity, you will find that in terms of obesity within the adipose tissue. If you induce obesity in mice, after 2 weeks of high-fat diet, you will find telomere shortening only in endothelial cells or vessels that are vascularizing the adipose tissue. They're quite impressive, okay? We don't know exactly how, why, but I think that studies are still ongoing, but this is something interesting.
And I also think, because from here, they said that, like, one of the things that they said, which I thought was interesting and links, is that they studied the tissue samples rather than the specific vascular cell types. So the data that they got was based on the tissue, and I think that was one of the limitations for the cell types. Yeah.
Of course, because here we have to understand that they are measuring the impact of all these things, but we can also imagine, and this is something that is suggested by the nitric oxide measurement, that small muscle cells can be affected by the mutation, by the deletion of TRF2 on endothelial cells. But finally, because you have to know that endothelial cells, but I think that you should know that, are promoting vasodilatation by producing nitric oxide that is diffusing to the small muscle cells that permit the vasodilatation. Okay, so in this context, it's quite important and interesting to, of course, to identify, of course, the cells that could be at the origin of this induction of telomere shortening. Samuela, I guess you had a question before, or maybe your sister Emanuela, I don't remember.
No, it's okay. I had a question.
Yeah. So maybe it was because someone raised a hand, that's why.
It was me.
Okay, so sorry for that.
No, it's okay.
On this, yeah?
I was gonna ask the question if that's okay.
Okay.
Thank you so much for your presentation, Janine and Isabella. I wanted to know how might other cell types, also known as like the smooth muscle cells, how can they contribute to the phenotype?
I think I can answer this question. So you're asking about the different like cell types such as smooth muscles would maybe contribute to what we're seeing in the study?
Yes.
Okay, so in the context of what the study looked at, we have a few different cell types we could look at, such as like smooth muscles, endothelial cell interactions, or immune cells. So for example, with smooth muscles, they line the blood vessels and they're essential for maintaining the vascular tone of the vessels. And when these smooth muscles actually undergo senescence due to telomere capping, they can use contractility, which can affect the blood vessel dilation and contribute to higher blood pressure. In the case of endothelial cell interactions, when endothelial cells are senescent, they release inflammatory signals, which can also induce this activative stress that we're seeing. And this can also actually influence the smooth muscle cells and therefore, again, compounding this vascular dysfunction that we're seeing. And then in the context of immune cells, when we have inflammatory immune cells, in the case of senescence, if it's influenced by telomere dysfunction, this could actually infiltrate the vasculature and might exacerbate this endothelial damage that we observe. And again, enhancing the stage phenotype. So all of them kind of play different roles, but like compensatory roles or collective roles on inducing this aging phenotype that is observed.
Thank you so much.
Yes, of course. So today, the third day, so we are starting by telomere. We can also start by inflammation. We don't know exactly who starts first. We know that, of course, this is, and senescence can be also resumed to kind of multi-domain triggering, because this is something that could start with inflammation. We're going to see that with metabolism and balance, or here by DNA damage finally. But this is something that should be approached by different view, of course, and today just one view. I have just another question based on the question that I've been asked in the audience about how can I say that? They are focusing on at the last part of the publication. They are focusing on a kind of oxidative stress. Do you know why they did that? And what is the relationship between oxidative stress and TRF2 finally?
Can you repeat the last part? Oxidative stress and?
Yeah, why is they did that? Why is they investigated? Why is they investigated the relationship?
Oxidative stress?
Yeah, and the deletion of TRF2. How do you make the link between both?
So I can answer maybe, and then Bella can go next.
Yeah.
Okay. So for me, the reason why they looked at oxidative stress was because senescence is directly linked to oxidative stress, the reactive oxygen species, and what they wanted to do was see if ROS was a key player, a key molecule for maintaining the vascular tone. And that's why they wanted to see its relationship with TRF2, because the one of their key not their aim, but one of their aim was basically to show that TRF2 deletion induces telomere uncapping, but they wanted to show a casual link, the relationship between oxidative stress and senescence signaling. And what I understood was the model needed to wanted to show a casual link between telomere dysfunction and age-related vascular changes.
Yeah, okay. That's because you just, Mariam, your microphone is on. Can you switch it up, please? Mariam, do you hear us? Yeah? Mariam? Maybe it's not your name. It's called Mariam, right? Yeah? So Mariam, if you can switch it off, that could be great for us because we hear you. So just, Jenny, to jump on your answer, I agree that I understood that they did that because, of course, we can make a kind of transition between telomere shortening and senescence. But finally, just to be on the same page, and this is something that we're going to develop to the metabolic or metabolism workshop. We are basically think that metabolism alteration leads to senescence, okay? And we are not used to think about the opposite. It means that telomere shortening should also, feed finally a kind of oxidative stress. You know what I mean? So this is not a unilateral relationship. You can find also through bilateral regulation some counter-regulation because, of course, when you are altering your cells, and this is a case here by mutating or by deleting TRF2, you will have some associated features such as an increasing aging process that could also affect some other process such as metabolism and in particular oxidative stress because this enzyme are highly sensitive to a particular aging, right? So here you have to keep in mind that even if in the past we discussed about the relationship between senescence and what we call the metabolic senescence, finally here we are trying also to understand why if the primum novens, if the first event was to shorten the telomere, and we're going to come back on that, we can have some associated feature that could affect also other parts of the global aging, such as oxidative stress. Okay.
Yeah, I agree, but I also feel like this was a proof of concept study. I'm not sure how far, because I understand like the reversal, but then I would also think P53 because they go straight to P21. So I feel like you can't really, it's more like a proof of concept study because they're not looking at other markers.
Yeah, I agree. I agree.
Okay.
I agree. I have another question, but it's the kind of comment and not really a question. I would like to know if everybody in the room is okay with TRF2. Do you know what is TRF2 or not at all? You never heard about that. Okay. I don't know if you have a scheme. I have one of in my computer, but I don't have memory stick. Can you just share me a memory stick if you have it or not? Nuclear USB. Nuclear USB. I can share you, but this is not very important. The drop on. Yeah, thank you very much. About you know what it is, the telosome or telotome? Did you hear about that? About this protein structure, or you never heard about that? You guys in the room never heard about telosome, telotome? Do you know how telomeres are produced? By which structure? Do you know? No, never. You never heard about telomere, telosome, or something like that? I'm going to show you. Wait 1 minute. Because I think it's quite important for you to know exactly what we are doing. And to see at least once. So you have to change. My computer doesn't recognize you. Something. Okay. You can ask questions also to your colleague if you want during I do that. Do we have another question? I hope we have another questions. I cannot be satisfied by only one question, you know. So I think that you should have some comment or some question. What are the question you can ask to the comment at least?
Yeah, there's someone in the chat ask the question.
Oh, Janine, Emanuela, sorry for your presentation. Criteria 2 deletion without telomere shortening still leads to dysfunction. Thank you, Emanuela.
Okay,
So
I think I can answer this question. So thanks, Emanuela, for the question. And so even though there isn't telomere shortening, the reason that there is still dysfunction could be for a few reasons. Maybe structural instability, because TRF2 actually maintains what's called the structural cap of the telomeres, which prevents the chromosome, or protects, should I say, the chromosome ends from being recognized as DNA breaks. And when TRF2 is deleted, these telomeres are actually uncapped, so that protective cap is no longer there, which then activates the DNA damage response, even though the telomere length might still be the same. Another reason is the activation of senescence. So when a telomere is uncapped, it actually signals for P53 or P21 activation, which then triggers the cellular senescence pathways and inflammation, which again occurs independent of the telomere length. And then another reason is also an increased oxidative stress, because the study actually showed that when the telomeres are uncapped, there's an increase or elevation in reactive oxygen species, which we know degrades nitric oxide and therefore impairs the vascular relaxation and contributes to this dysfunction as well.
Is it okay for you, Emanuela?
Yeah, it's all good. Thank you so much.
Okay. Do you see the slide here with the telomeres in line? Yes. So just a few words about that, just for you to identify. So this is exactly what is the cultural structure that is able, but it is the origin of the synthesis of telomere. So just to be sure that you got it, I don't know if the previous one, you know, I don't remember if you already saw that, maybe in Master 1, because you come from here, for most of you. And I explained to you what was exactly telomere, that is non-coding DNA that is added at the end of the chromosome, and that is a kind of security system. But here, what is interesting is that today we are focusing on this structure, this protein structure, which is compounded by different proteins, TRF1, TRF2, TIM1, TIM2, TPP1, etc. And you can see here that all the proteins should have their role in the capping or uncapping. So this is one of my open questions I will ask to Janine and to Isabella. Can you just re-explain what is really uncapping, what you call uncapping, and what is uncapping in the publication, please?
Yeah, I can go.
Yep.
Yeah, okay. So telomere cap chromosome ends, and this protects them from DNA damage. But the loss of the telomere protection, which is uncapping, it activates senescence, triggering inflammatory response. So that results in the senescent cells releasing ROS and reducing nitrogen oxide. And in the context of this paper, it would increase vascular stiffness and the risk of hypertension. And yeah, that was basically what I had.
That's a good explanation. I thank you. Is it okay for you guys? Did you understood? Did you understand? Sorry, what was in capping? It's crucial for you. This is something, for instance, this kind of question I can ask you for the exam. What is the definition of telomere and capping? Okay. Yep.
Is the uncapping part only happen in senescent cells, or it can happen in non-senescent cells?
Well, it can happen. No, I mean, that's uncapping should trigger one. That should trigger a kind of process that will lead to senescence.
Okay.
That means that, but senescence is not only due to uncapping. Okay, senescence can be due to other things that telomere shortening or uncapping or whatever. So that means that if you have DNA damage, such as, I don't know, mutation or whatever, you can have senescence without any effect on telomere shortening or even uncapping. Okay, it's quite important for you, for all of you, to understand that telomere shortening, it cannot be the only reason that promotes senescence. For instance, you should maybe know that in mice, the telomere are very, very long. So long that we consider that during the lifespan of the mice, it's quite difficult to start to degrade the coding DNA. In humans, that's quite different. They are shorter at least. And we know that at the end of the life, of course, when you age, you are going to induce some mutation or some DNA alteration because of the length of your telomere. Okay? So this is quite important to give the proof of concept, and that's a good thing that we start with this publication to see that if we are altering, modifying the telosome, so TRF2 in particular, we can also, I don't know, target TRF1 or TP2 or something like that. We are going to induce a kind of accelerated aging. And this is exactly what they saw. They focus on endothelial cells and particularly on vessels. But we can imagine that we can do that on different tissues, such as, I don't know, the liver, the brain, etc., etc. It has been done after this kind of publication. And we can easily see that if you are altering, of course, the telomere, the telomere synthesis, of course, you are going to promote an accelerated age. Okay. So I have other questions or the comments, maybe to add to the discussion. So I have one that has been answered by my scheme, other protein that could be involved in uncapping. So you can consult this kind of scheme that you find. Someone is on the door, no? You can open. I have a question, maybe an open question to the room, and because it's not only Janine and Isabella that have to answer, maybe what is your opinion about the use of this inducible model? Did you understand the model they use? So they use a Krelox model. You know what it is, I guess, I hope. And this construction is inducible by the treatment with tamoxifen. You know what it is? This kind of thing? Okay. Why? Why did they do that? What is the reason here? To focus on this tissue could be one reason when you do something like, which is, of course, under the dependency of a promoter. Here is not the space that I want to control, is the time. Yes. So it's a control of the time. You can induce the deletion whenever you want. Why?
If I recall correctly, like sometimes there is an in vivo model where they use tamoxifen in order to knock out certain genes.
Mm-hmm.
So maybe that's why like they use in order to knock out the gene coding for this.
So what would be the consequence if not, if they are not using this inducible model?
They should use another model.
No. Yeah, so maybe I didn't, can you say it again?
Yeah.
To be sure that I well understood?
Yeah, I mean like because this model was induced by transplant.
Yeah, that's it.
So they use it in order to knock out the gene coding for the TRF2.
Yeah.
However, if they did not use tamoxifen, they should use another model, for example, using like an animal without the gene or something like
What
that.
is the consequence of this model? That's why I asked the question.
Yeah.
It's a question of lethality, I guess, of course. I mean that you can choose to make the total knockout and say that, okay, I don't care about the time and space control. What I want to do is just to delete the TRF2 gene. And if you do that, of course, and obviously, I guess, that you will have lethality because TRF2 is crucial, of course, for the embryology, etc. So that's why, according to me, they choose to make an inducible strategy. And this inducible strategy is to wait that the mice are adult and to inject tamoxifen to induce the mutation. They have a results, I guess it has been presented by Janine at the beginning, about a loss of, how much loss? How much is it? Something like 80%, I don't remember.
85.
85%, okay, this is something that could be considered something interesting, even if it's not a total absence of the expression. It was checked by what expression of the gene, of the protein, I don't remember.
mRNA in the artery.
mRNA expression, so yeah, mRNA expression is not perfect, but this is something that we can consider something acceptable, of course. Well, yeah, I have a special question here again. That is a question, an open question for the audience. What do you think about linking here in this condition the telomere shortening in one hand and how can I say that, the cardiovascular, the reduction of vasodilatation? What is your intellectual process that can link these two different things? How can you connect? It has been maybe mentioned, I guess, by Janine before. What I would like you to answer or to discuss, it's of course your point of view, but maybe you heard about some specific feature that happen with age in vascular organs. What do you think about that? What's happened when we age? Did you have some course about vascular aging? I guess that at least people from VUSG had. So sorry, but what is the main characteristic of an age vessel? Janine, maybe you can help because you pronounced this word before about what's happened when we age within the vessels.
The primary risk factor for cardiovascular disease?
Yeah, it is. But what I want to know is what is the kind of which process?
Like angiogenesis.
Like
Angiogenesis.
no, angiogenesis is something that can happen even when you are young.
Adaptation.
Yeah, and what is this adaptation? What about this adaptation? What's happened? What's happened when we age? Did you already ask the question why we are maybe more prone to vascular disease when we age? Why?
Stiffness is
Yeah, that's the key word, the stiffness. We realize by more and more publication that when you are considering the vessels and the vascular system of rich people, there is an increase of stiffness, okay? And this increase of stiffness could also maybe explain why you have a compensatory effect on heart. So that's why, you have to, and the stiffness, c'est la rigidité, okay? I can see you. Don't hesitate to raise your hand. I don't understand this word, and I am here for that. So where I was, yeah. So the stiffness you have to consider, like, if your vessels are not able to be flexible, of course, at the end the heart will compensate. So that means that the heart must pump more and contract more, etc., with a high frequency. So that's why finally we can explain that during aging, you have this kind of sequence of events. But at the beginning, we can ask the question why there is this stiffness. Yeah.
connected with
That's the next question. Why there is this stiffness? Is it connected with maybe atherosclerosis? Is it something that is linked to, I don't know, some, you know what are the rages? Like, you know, ages, like, how can I say that? The modification of proteins that are circulating proteins that are maybe polyglutaminated or with different lipids, sorry, that could impact the quality of the vascular wall. So this is quite important, and a lot of people are trying to understand why there is an increase of stiffness. Sorry. On the other hand, you have to know also that endothelium is a tissue that is able to regenerate. Okay, when you are young, your endothelium are regenerated. Unfortunately, like other tissues, when you age, the endothelium regeneration is lower, of course, and you have this consequence of age endothelial cells, that couldn't maybe promote what we are waiting for them, which is synthesis of nitric oxide and all the parts that are able to play on vasoconstriction and vasodilatation. You should know that a vessel, I'm just drawing a vessel for people that are in the room. You should know that a vessel is something like that, no? With the endothelial cells. You know that? Okay. And surrounding that, you have the small muscle cells. And what's happening when you need to make a kind of vasodilatation? These endothelial cells are going to produce nitric oxide that is diffusing here in order to promote the vasodilation and vasodilatation. Okay, so what we think, and if you read the literature regarding the explanation of maybe stiffness, is first the incapacity of these endothelial cells to promote the production of this kind of factors. That's why in the paper presented by Janine and Isabella, they are trying to understand exactly what is the consequence of NOx, but not also. And then this second explanation, do you have an idea why this stiffness is here? Yeah?
Hypertension.
Yeah, that's a consequence. Hypertension is a consequence of this vascular stiffness. But why at the cellular level?
Maybe the muscular cells less react.
We can also imagine that the small muscle cells, of course, are maybe less react. But you know, when you are thinking about a kind of tissue that can be sclerotic, what is your conclusion? What is responsible for this kind of tissue sclerosis? You know what is tissue sclerosis? It's when the tissue is kind of scared. Okay, so what do you think about that? Which compound can be at the origin of this kind of processes? Yeah.
Fibrosis?
Yeah, the fibrosis, but the fibrosis is due to, what is fibrosis finally?
The axonal matrix.
The
axonal matrix when it's really so.
Yeah, that's it.
Not like
If I understood it again,
Yeah.
that's it.
the ECM.
Yeah, the ECM. So you know what is ECM, extracellular matrix? So these cells, we don't know exactly why when we are more age, but you can also generate to a lot of different cells. They are synthesizing more extracellular matrix, accumulation of collagen within the cell and of course outside the cells also. And the collagen is going to accumulate and then participate into a fibrosis process, sclerotic process, which can be also at the origin of the stiffness of this vessel. Okay, so you have different parameters. And at the origin, you can maybe think that because these cells are affected by, I don't know exactly what, but the telomere shortening of the cell is going to induce this kind of different features. Okay? Is it okay for everyone here for this part of stiffness? Very important stuff that you should be able to at least to explain. Yeah, I have a question for you, Janine, maybe the last one. I don't know if I hope that in the audience they have also a question. Can you explain just what is the RBC fraction? I guess it was Isabella when you present what is RBC fraction. Because you compare micro vessels and between maybe other vessels, and I'm not sure that you call it because you go very fast on that. You went very fast on that, and I would like you maybe to come back on this RBC fraction.
RBC.
Was you?
I think it was Bella.
Was it Isabella? You know what it is or no? I don't remember. That's why.
Yeah.
To be totally honest, I don't remember what they are measuring here.
And I'm trying to find the slide with it.
It's a figure 4, number A. Figure 4 for the A. The RBC fraction, microvessel diameter, but I don't remember how do they do that. It's a kind of perfusion stuff, no? If I remember. Isabella, do you have something about on that or no?
Oh, where they looked at the microvascular perfusion.
Yeah, that's it. This is a kind of perfusion measurement, no?
Yeah, they looked at the red blood cell fraction across a range of different mesenteric microvesicle diameters. And I think the reason they did that was because obviously the diameter affects the perfusion. So they wanted to measure, like, is the perfusion affected by the TRF2 deletion, specifically in microvesicles.
Okay, thank you very much. This is what I wanted to know because I didn't remember. It was just for me. Other question, other comment. What do you think about this publication? What do you think about the relationship between closome, TRF2, senescence, associated aging? I don't know. What do you want to discuss? Is it okay for you if I have some question about this paper? It will be good for you? All of you? Yeah, you sure?
Yeah.
For you, I hope, Gavin. Also to the audience because they are a little bit shy. Now, today you don't have to be shy. You have to discuss, to open some stuff in your head. Okay. But sorry, but I was guessing that telomere was that or that. Okay. So today is your day. So that means that you have to raise the question and to ask the good stuff, maybe to better understand and to be sure that you're on the good way. And if English is a problem, here again, I repeat and repeat and again and again. So don't hesitate to raise your hand. Can I ask a question in French? Because I didn't understand at all. So maybe we can switch to other explanation. Okay?
I have a question. Maybe you can help me understand because I see in the paper that they looked at P21 and they're showing their results for P21. They're showing their results for DAPI and their Western blot as well. But why weren't they showing their results for P53? Because P53, as I understand it, is at the top and then is P21 activation. So I know they did like a whole experiment to rule out P53, but I was wondering what would be one of the reasons they wouldn't show the result.
Well, I don't know if they I mean, that if they did, do you remember if they did that? They did, but they didn't want to show. They said that not shown results, right? Or something like that?
Yeah, they say like they did the experiment with P53 to rule out the influence of it. But they're not showing any of the results with P53. Yeah,
in that case, it can happen that maybe if you, so two different reasons. The first one is because that these two parameters are complementary. So when you have activation of one, it was just an argument to measure the senescence activation for them. So you can measure P16, P21, P53. Of course, that you just explained that they are not both on the same pathway. But at the end, you can measure senescence like this way. The other reason, because of course, I don't know about the editorial line of this journal, but they can also, it was the first reason, because maybe they asked them not to repeat, different measurement of the same question. But also the other one is to say that if I remember, P21 was measured by histology on the slides, right? It was on figure 2. Yeah. So maybe the histology were not good enough to be presented, and the quantification was not very beautiful, and the Western blot also. So they prefer maybe to present at least P21 and said that they did also that for P53. They frankly think that P53 is also activated, but they prefer not to show it. This is just between us that I can say that, of course. It depends on the reviewer. If the reviewer asks you to do P21, P16, and P53, and to present it in the world figure, you do that. In that case, I think that P21 was enough. Okay? Other question, other commentary, what do you think about that? You want to go to switch publication? Okay? Okay, so let's do maybe another one which is dedicated to the measurement of telomere. So the two resting publications are more complicated to understand, okay? So that's why we're gonna try to do that step by step, because there are, I mean, that's something like 2 or 3 figures per paper, so they are shorter, but they are maybe more difficult to understand. What I wanted you to do, and I see your face because you are a little bit stressed by that, don't be stressed. I just wanted you to do and to show to your colleague how we were supposed to measure the telomere length. That's it. I would like you today to come back to your home with the knowledge on how we can measure the telomere length. And I will ask you the question, so be prepared to that. What do you think about what is the next experiment we can do in the future, maybe to evaluate the telomere length? What is your opinion, just for discussion? Okay? Who wants to start? Which group? Okay? Can you come here, put some memory stick here? And so thank you very much again for your presentation, Janine and Isabella. You did really well. And we're gonna continue. Thank you very much. N'hésitez pas à poser des questions, même en français, je traduirai, ne vous inquiétez pas. Vraiment. C'est votre intervention. Hein, si vous galérez, si vous ne comprenez rien à ce qu'on dit, vous parlez en français. Ça fait. D'accord? Soyez pas stressés, parce que. Vous taquez, ouais, ça met un peu de temps. Hein. Ça met un peu de temps. Ça met un peu de temps tout à l'heure. Ou alors tout un peu le truc, là, tout à l'heure, j'ai tout fait là. Ouais, j'ai fait un truc là. Mais ça le fait plus. Ça en a 2. Pareil pour les présentations, je le dis en français, hein, je le redis en anglais. Ne soyez pas stressés. Le but, on n'est pas là. Même si je me mets là, c'est pas le but, c'est pas de vous évaluer, là, on s'en fiche. Bon, par contre, il faut la faire avec un peu de sérieux. D'accord, la présentation, vous soyez posé quelques questions. Si vous l'avez préparé demi-heure avant, c'est sûr que ça va être compliqué pour vous, pour tout le monde. Voilà. Donc, il faut que vous la fassiez avec sérieux et que vous sachiez à peu près, voilà, quoi dire. Pour ceux qui ont des revues, vous pouvez choisir aussi de le présenter tout ou partie. Parce qu'il y a 2 revues, je crois, dans les prochains jours. Et donc, vous pouvez choisir de le présenter tout ou partie. OK? Oui? Donc, nous avons Apolline et Farah, c'est ça?
Yes.
On peut vous interrompre ou parce qu'en ligne, tout à l'heure, on n'a pas interrompu, mais est-ce que c'est mieux qu'on vous interrompe, vous les faire tout d'un coup et we interrupt you, ou vous parlez toutes les 2 français ou non? Speak French? C'est quoi ce que je veux dire, speak French? Donc, on fait quoi? Maintenant, à peine? Si vous êtes stressé et que vous ne voulez pas qu'on vous aide.
On attend la fin.
On attend la fin? Allez, on attend la fin. Et par contre, vous notez les questions, vous avez à leur poser. Parce que sinon, si je suis tout seul à parler, ça n'a pas d'intérêt. Moi, je les connais, les papiers. Alors, on écoute Farah, ensuite Apolline, ou pas Apolline et Farah.
Hello, we will introduce.
Vous le partagez? Il est partagé avec
Je ne suis pas sûre.
Non, alors Farah, là, tu peux appuyer sur Escape, là. Voilà. Non, c'est sur là. Et
Pas sur la. Oui,
ouais.
lui là.
Oui. Ah, oui, ça va. Il va aller.
Sur la vidéo directement.
Vas-y, Daniel.
Le truc de droite. Oui, c'est ça, c'est le truc de droite. À droite. À droite, c'est ça.
Alors, encore.
À droite,
Là. Là, partagé. Et tu l'as là. Ça fait. Ouais, c'est vrai qu'il y en a qui sont pas mal. It's okay for you online? You can see the April and Farah presentation? Give me a yes. Give me a sign.
See it,
Thank
yeah.
you. Okay, go ahead.
So hello, we introduce today the article. The article entitled "Extensive Allelic Variation and Ultra-Short Telomeres in Senescent Human Cells" for Ducans and in Africa. So we will begin with the introduction, then method and results, and we will finish by discussion and some perspectives. So to begin, what are telomeres? So telomeres are a specialized structure at the end of the chromosome, and that contains hundreds to thousands tandem repeat of the sequence of TTA-JJJ. So these telomeres are shortening with each cell division, and eventually this shortening will trigger the irreversible arrest of proliferation, which is called cellular senescence, which lead to aging. So there is several mechanisms of telomere erosion, such as variations in the end replication problem, replication slippage, and oxidative damage. So likewise, various models have been proposed to explain how telomere shortening triggers cell cycle arrest. So it is so the consensus, the current consensus is that the signal is generated when telomere length reaches a threshold of 2 kilobases. So this leads to alteration in the binding telomere associated proteins, which lead also to a conformational change at the telomere. And this suggests a regulatory mechanism. So to test this mechanism, critical testing of these models requires the ability to record the full spectrum of individual telomere lengths at a single chromosome terminus in both dividing and non-dividing cells. So there is two methods. The first one is TRF, terminal restriction fragment analysis, which uses restriction enzyme digestion of genomic DNA, followed by a sudden hybridization to a probe containing a terminal repeat to measure the average size of the TRF for all chromosomes and simultaneously. But this method has some limits, with like a low overall sensitivity. So it's it needs at least 1 microgram of DNA for more than 10 exponential 5 cells. And also it is based against the detection of short telomeres. The other method is a QFISH for quantitative fluorescence in situ hybridization, so this technique measures the fluorescence arising from the hybridization of TTA-JJJ enzymes in situ to metaphase the chromosomes. These measurements of the chromosome-specific telomere lengths, and this allow us to detect the chromosome termini with little of, if any, TTA-JJJ. But this technique has also some limits, such small data sets can be produced, and here we require metaphase chromosomes, which restricts analysis to cells that are still proliferating. So in this paper, researchers have developed a new method named Stella for single telomere length analysis, which is a PCR-based approach that accurately measures the full spectrum of telomere lengths from individual chromosomes. So this analysis, so in this paper, we will analyze the telomere at the end of the short arms of the human sex chromosomes, XP, YP telomere. And this will identify several features of telomere biology. So the first step of STELA is the first step is consists of annealing a linker tellurate. This step. So annealing a linker named tellurate, comprising seven bases of TTAJJJ homology, followed by a 20 nucleotide non-complementary tail to the J-rich 3' overhang of the DNA or of the telomere. Then in the second step, so we will ligate this tellurate to the 5' strand end of the complementary J-rich strand of the chromosome, which effectively attacks the end of the telomere with the non-complementary tellurate tail. And then the final step is PCR. So now PCR can be performed with the telltale, which is the primer, that is identical to this tail, and in the same time, in the other part of the chromosome, with the chromosome-specific upstream primer located in the third telomeric region, which also can be made allele-specific if necessary by exploiting known polymorphism in this region.
Thank you very much.
Sorry for my English, I'm going to read. First, on the left, we see the results of TRF analysis with 1.5 micrograms of DNA from 2 clones of the K1 human thyroid cancer cell line with mean TRF value of 2.9 kilobars and 1.5 kilobars respectively. By using the STELA method, the STELA method use 400 micrograms of DNA from the 2 same human thyroid cancer subclones, in which reaction A and reaction B with tellurate 2 ligated. We see the results. For the reaction C, D, E, F, G, they are a negative control. Stellar products are not generated in the absence, for example, of the 3-terminal overhang, or if the linker is irrelevant for the ligation. On the right, on serial dilution, TRF like a Smearson with the highest input DNA is eventually result in 2 single bed, consists with the detection of 1 single telomeric molecules. For the first, the study validates the technique of STELA by showing that STELA products of a size consistent with TRF analysis are generated after ligation with the tellurate liquor. This observation suggests that the PCR amplification is highly telomeric specific. There is a graphical representation of allele-specific STELA data from young MRC5 cells with only 29 cell division and MRC5 senescent cell with 55 cells division. What we see, so we see, we found a bimodal distribution. Single allele amplification in the fibroblast strain are heterogeneous with the respect of the polymorphism shown by the distribution. The distribution are allele specific and heterogeneous with sometimes 6 kilobase of length difference. Now there is an analysis of a one pedigree with one mother and 2 sons. This analysis of one pedigree shows that the XP-AP telomere length at senescence is inherited in a non-Mendelian fashion. Indeed, we can see one allele. Indeed, we see that there is a stochastic element in a telomere inherence. For example, here we see that there is no AT and just DC. After they try a new analysis, they use Stella to examine telomere repeat dynamics during in vitro culture of primary human fibroblasts. At senescence, the mean of the shorter alleles varied between 6.2 kilobars, and the value are lower than previous estimate based on TLF analysis, for which the mean is between 4 and 7 kilobars at senescence. For example, the mean of the lower MRC5 allele decreased from before 3.8 kilobars to now 1.2 kilobars at senescence. We can also see that some telomeres were devoid of telomere repeats at senescence with under 45 kilobars. So that indicated that TTAGGG loss occurred from a subset of chromosomes at senescence, and it's in contradiction with previous current models. Last, we can observe occasional bends in both clonal and non-clonal culture that are significantly different from the bulk distribution. This may reflect replication stage, telomere recommunization, or oxidative damage. To conclude, first, the method Stella single telomere length analysis is a PCR-based approach which provides more single telomere molecule data by analyzing the full spectrum of telomere length from individual chromosomes. It was used for the sex chromosome, but it can maybe be used for other chromosomes. we can note that it's need labor intensive, and that the stela cannot be used for a longer telomere. Compared to the current method TRF assay, stela is developed to overcome several problems in detected abrupt telomere shortening in a single chromosome. For the analysis of XP-AP telomere in fibroblasts, we can note different things. First, we can note bimodal distribution of telomere in normal fibroblasts, which means that there is an extensive allelic variation which is maintained for the development. We can also note that the rate of erosion are independent of allele size. And last, we can note that some allele show almost complete loss of telomere repeats at senescence. This method shows ultra-short telomere in senescent human cells. So this discuss the fact that telomeres can be used as an universal clock for aging.
Thank you very much.
It's okay.
It's not finished?
It's just
I don't know, because you were looking at us.
no, it's my fault.
Wanted just to upload, but
Just the use of this stela method with a date are published in the magazines. The stela method, for example, was used for the response to chemotherapy in cancer, and this method was also used to mark to the agent.
yeah. So that I told you that the second one and the other one were more difficult to understand, of course. And I think that you did a great job because it's quite difficult. Well, first of all, before we are starting discussion, I think that what we need to keep maybe, and this is, thank you for doing that, is the table you put, at the end of the both presentation, at the end of the Farah presentation, and at the end of the presentation. First of all, thank you very much for the introduction. I think it's quite important to get the background. And for this, it was really important for you to see what is its TRF and what is Qfish, to compare that to the stela technique. Here you understood that we are on the technological level. We want to understand exactly how does it work, how we can measure this telomere length, etc., etc. So it was a little bit difficult when you are not an expert. I'm not an expert to understand this kind of paper. I don't know what is your feeling. What did you feel about this paper?
It was difficult.
Yeah, really? Why? By the language used, by the different, by the technology, by what? The technology are not so difficult to understand. What is can be different or can be difficult to understand is the analysis of the different gel, etc., and the use of different models. So you have any question before I can ask mine? I'm going to come back with you. Yeah. Question, comments, whatever you want. In French, in English, in other languages.
I'm going to try in English.
That's right.
I have a question about these techniques. Would the stela techniques used to monitor telomere length during, for example, antiretroviral therapy known to des raccourcissements de telomère.
To use some telomere shortening.
And enabling direct monitoring and adjustment about adapté à chaque personne.
So I love the name of the question. So you want to know if this can be adapted to cancer cell, if I understood, and if it's something that could be adapted to maybe monitoring the telomere shortening in case of cancer cell. That's it. In case of inducing cancer cell. So this is that what you present at the end of the presentation.
It's not to follow, it's to predict. I don't see a publication about follow-up of telomere shortening during therapy.
What do you mean? Do you mean that it's not to follow, but to predict? That means that you cannot. We are just going to make some cross-sectional study and not longitudinal study on the same cell or the same station.
I don't see article
Okay.
Publication about the follow-up.
So that means that regarding the layer question, this is not something that could be associated to kind of marker or to kind of follow-up mark. I don't know what you want to say. Follow-up marker, you know what I mean? Marqueur, un marqueur de suivi de situation.
It's just not done.
Yeah.
But it's not impossible.
Because that means that this is not done in real time. We cannot follow up telomere shortening in real time. We have to kill the cells, of course, each hour or each 10 hours. I don't know what is exactly the question raised by there about the kinetics. But if we want to just follow up to say, okay, I would like to know if at this point, at this time point, after tamoxifen treatment or whatever, I can see the telomere shortening. You think that this is not something doable because, of course, we have to kill the cells to do that. So we cannot follow it up, right?
Yes.
And do you think that in the future we'll be able to follow in real time the length of our telomeres? Can we imagine that in the future, did you see something around that? It's right also for other groups that are dedicated to the third paper, and of course for you guys.
In vivo, you mean?
I would love, yeah, by an appli on my iPhone. I would love to know exactly what is the length of my telomere just by, I don't know, kind of a blood withdrawal or something like that, blood analysis. Do you think that in the future we can be able to see or to know exactly about the telomere length? What do you think about that? Is it something that could be interesting also to get this information or no? Do you think that telomere length should be, how can I tell that, affected in a kind of real-time manner?
Like, what is the significance if you actually determine how this would have this? You know, I think aging.
Yeah, but telomere length we just explained with the first presentation in this one. This is the long-term process, right? I mean, Farah just presented us that this is something that is done to replication. We are losing some materials, etc. So, of course, for cells that are highly replicated, like I don't know, skin or something, skin cells, or some epithelium, such as gastric epithelium, etc., that could be interesting to get a kind of real time. But at the end, if you will, I mean, that if you try to analyze this at different time points, what I agree with you, Apolline, is that maybe what is interesting, or with the question raised by Léa in the cancer, is to see the dynamic. You know, to see the dynamic. That means that are we losing some telomere length, you know, from the last 5 years, and are we losing more than it can be expected? So that means that, of course, I'm going to turn pathologic and to turn senescent because the slope of the loss of my telomere material is too fast compared to the global population. That could be the, of course, this is not something global right now. We still the difficulty to analyze the telomere length. Okay. Other question, other remark? Yeah, again, or maybe.
I have a question.
It's okay. We are just discussing. It's not a question answer. It's discussion.
Is there critical threshold judging to know when it is considered critical?
Yeah.
Or I didn't understand you.
What about
There was
the vision?
narrative.
The threshold is estimated at 2 kilobases. And
But you mean about what? 2 kilobases is what? Is the loss or is
the length.
the length of what? Of the
The length of-
telomere?
Yes.
After that,
After
the
that-
DNA, the coding DNA is in danger. We can say that. It's
Yes,
something that can be altered.
Senescence.
Wow.
It's,
I didn't know this value about 2 kb. I learned a lot of things during the presentation. Really, thank you, because I learned a lot of things. I didn't know this value. This is something that has been
From the last article,
recently, or this is something that.
it is old.
And this threshold is not related to different patient. I don't know, to the sex or to the gender.
No, it's general.
It's a general.
Men, women is the same. And even and regarding the question raised by Leah, if I have a cancer, do we consider that this threshold also is something that is acceptable?
If I understand good, they show in this analysis that some cells are under threshold during senescence or not.
Okay, I didn't know that, really. Really, I didn't remember, I didn't analyze it like that, and the 2 kb value, I think I wrote it, so that could be something that could that I could ask you, of course, in the
For
future.
this one, we see that the
Yeah.
area C5 is under
Yeah.
2 lobas
Yeah.
special,
But
but no, not but other are under.
yeah, but you mentioned something about the MRC5 cell.
It's just an example to see that the
So this guy is under the 2 kb considered as a kind of, it should turn senescent. All these cells that are under the 2 kb value should turn senescent in the future. We should.
no, they are all at senescence, and
Yeah.
we see that for some or others, the
Okay.
the length.
Oh, it's still not senescent. Yeah, I understand what you mean. You mean that we can reach the senescence with different time points, with different points. Right?
But
We can reach the senescence, but with different points of telomere length. For instance, this guy are entering into senescence even if the length of the telomere is 7 kb.
Yes.
So these cells are cancer cells. IMR 90, IMR 91 are cancer cells. The other one, I don't know what W38 are, I guess, are normal fibroblasts, I guess, if I remember. But these guys are cancer cells.
Yes, and the 2 cancer cells don't have allelic difference.
What is the allelic difference for you?
Is difference of length of allele. Sorry.
So we have 2 alleles,
Yes.
okay, and that means that one is different from the other one.
Yes.
So it means that it's a bad sign of loss of uncontrolled loss of telomere length. That's it. We are maybe losing more telomere in one allele than in another one. Okay. Go ahead. You wanted to present that?
No, it is about the inherence of the allele length.
And what does that mean here? Can you just say it again? What is
The
it?
mother has
So for those that didn't understand, the mother is here, okay, with the here, and the 2 sons are here. Okay, so
The mother
just to make a kind of break here, we can see here that the lengths, so depending on the sequence, are not the same for the mother than for the son. Okay, and
no, that's it. The mother has 2 allele, and one has only 1, and the other has 2 allele too, and the lengths are not don't look like the length of their mother.
yeah, and what does that mean? The mother display some discrepancies, some differences between the length of the 2 alleles, right?
Yes, the mother, yes.
Yeah, and the son, for instance, the first one is not the case. That's it. It's not the case. We have the same length of the allele, but for the second sum, I mean that one allele is present, the other one seems to be absent, right? So what is the conclusion of that? You mentioned stochastic element in telomere inheritance. Remember or not?
I understand that one of the two alleles of CD, sorry, and one is
9 kilobase.
and the other one
Okay. So,
Yeah.
yeah. So that means that there is no kind of
correlation.
and this is non-Mendelian, the repartition. It's not because your mother is going to give you one allele that this allele is going to be a Mendelianly distributed between your allele. So that means that you can have some alleles that are very long and others that are very short. Is it a kind of is it a feature of bad prognostic to get some difference between the two alleles in terms of telomere length or no? Did you read that? If you come back to the cells, you know the cells and the first figure with the cells. No, just the figure comparing, you know, the telomere length and that for retinossenescence. Yeah, here. Is it something that is important when you have some difference? Is it something that is a kind of a bad prognostic for the cells, or we cannot link finally the difference between the two alleles with the occurrence of senescence? Do you know that or not?
I think so, because more the allele is long, more the senescence is.
And you're starting to degrade some DNA. So that means that even it depends, of course, of the stoichiometry of the expression of the different alleles, but normally they are expressed equally. And in that field, you can make you can lose some DNA. If the loss is it's a kind of mutation, you know. If the loss is predominant compared to the presence of the other one, that means that finally the protein will be synthesized. So you're losing some DNA, but it's not super important for the cell. In that case, it's okay. But in the case, of course, if you need the two alleles to promote the synthesis of the protein, of course you are deleting some stuff, and here you have some risk to get some cancer, to get some process of aging. Okay, so it's quite important. Do you have here again? Sorry, I don't want to get the floor.
Just to make sure I understood correctly, they look at the length of telomere here on different kind of cell type.
Yes.
And do you know if in a certain cell type, every chromosomes are shortened the same way or not?
Can you do it? Can you say it again? In these cell types, are the
in one
chromosomes?
specific cell type, is every other chromosomes are shortened the same way?
That's a good question. You understood?
No.
She wants to know if finally here they are measuring some telomere length, loss of telomere sequence in specific chromosomes, right? Yes. They are not doing all the chromosomes. If I well understood the question, she asked if we know if the loss we can see here for I don't know which chromosome it is something that can be generalized to all the chromosomes. That means that do you think that if I'm losing, and this is a global question, if I'm losing something like, and this is a question that can be raised for all of you, if I'm losing something like 50% of my telomere length, is it something that will affect all the chromosomes, all my chromosomes, in the same extent, 15% on one chromosome, or one chromosome that can be affected at 15 and the other one maybe at 1%? Do we know that? Do we analyze that? I'm not sure, but this is a good question. I have to say it again. I don't know. Maybe someone in the room has the answer. Do we know that if telomere, I mean that it should be risky if it's not the case. I think it's a global process. Because in that case, that means that, and that's the question of today, considering telomere length as a biomarker of aging should be forced. Because it depends on the chromosome you are measuring.
You can have a DNA mutation or something that happens to one chromosome.
Yeah.
and not the others and make a difference?
That's you are on the chromosome response. You are on the chromosome. What I mean is that you are on the capacity of the chromosomes to produce something. But we are not on the same thing. I was just thinking if maybe the telomere shortening was the same in the all chromosomes, and of course affecting maybe the number 10 more than the number 12, and the consequence will be more dramatic if you affect the 10 than the 12. Yeah. Want to add something?
Sorry, I'm not sure I all understand, but in this case, for different cells, they show that telomere of one subtype are very different than the mean of telomere length.
Yeah.
Because maybe of DNA damage or
Yeah, you explained that, and that was one of my questions, that this stain here can come from a kind of oxidative stress of DNA damage. Can you just tell me more about that, or tell us more? What do you consider, what do they say about that, about the fact that
it's not just telomere shortening,
yeah.
It's more radical, I don't know.
Yeah, say it in French if it's okay in English. It's not okay in English. But okay, I don't understand why. So that means that let's imagine that there is a lot of oxidative stress in this cell. It should impact maybe the telomere length, but differently
just differently that just by cell division.
Okay. Can you just explain for all of us, if you know it, of course, what are the corresponding steps in this kind of scheme? Because I see on the bottom, the kind of quantification of telomere length, but if you have to connect both graphs, what is where exactly? I mean, where are the young, where are the senescent?
Sorry.
Just for us that didn't read the publication.
It's great. The young and
okay, that the young PD19 and PD29 are the senescent induced cell. Okay.
Because of number of cell division.
Okay, I imagine. Yeah. What are the different tracks? What does that mean?
It's about the two different alleles for the group.
I understand the right and the left, but individually each track. What did they put on each track?
This one, for example.
Yeah, for example. Yeah, can you tell me, because I think that you could maybe don't know that.
Is a telomere length.
Here, this. What is the correspondence of each track?
Is the mean length. Mean length.
Yeah, but they're corresponding to what? To different cells, different. I don't know.
Amplification.
Yeah. So to amplification, but why they did that? I don't know, 10 times. You know what I mean?
No.
À quoi correspondent les différentes lignes? Pourquoi ils ont différentes lignes? Pourquoi ils n'ont pas fait qu'une seule ligne? Parce qu'il y a une seule cellule. Et chaque point, each time you are going to present the different deal here, you have a lot of lines. And do you know what is the correspondence of these lines? It's also the same if you compare the other one. We can understand that the molecular weight here. Okay, so here you are. This is something here, but why we have so much lines here, so much tracks?
C'est différentes pour les mêmes cellules, c'est différentes mesures pour avoir une mesure moyenne.
Et donc c'est ce que tu voulais dire moyenne, means, c'est ça que tu as dit tout à l'heure.
Oui.
Pardon, je sais pas. So this is exactly that. Yeah, of course, we are going different amplification, and you are measuring like a means, exactly what is the length of your telomere. So here, just if you can see this kind of scheme, of course, we are showing that for the young, we are mainly around 8 kb. For the young, we are mainly around 8 kb. You can see the distribution. For the senescent cells, there is a kind of shift on the left. Only for the GC alleles, you can see here you have the shift for the GC alleles. Is it also true for the AT? You have exactly the same thing. Of course, the telomere can be shorter. Here, you have only 1 to 2 kb. And regarding what has been said by Farah, 2 kb is kind of crucial limit. So we can ask the question if AT allele should be maybe more sensitive to the GC allele to measure the senescence or no. If we are losing maybe more, I don't know. It should be very interesting. We can see that the young one are 2 kb. So that means that here, this limit, I don't know if it's something that can be also considered as a premature aging. I think no, but
No, 2 kb is for the cell cycle arrest.
yeah. So that means that the cells will enter into senescence when we reach 2 kb. But I think that the 2 kb length.
It's apoptosis. It's the
Yeah, but before entering into apoptosis, these cells are going to transit through the senescence. So that means that they are going to enter senescence and then they are going to die. Unfortunately, during aging, these cells are going to be resistant to apoptosis. So that's why they accumulate during aging. And this is something we are going to discuss with the senescence workshop. Okay. Okay. Do you have any other question? I hope yes. Yeah.
I have just one question. If the critical length of telomere is 2 kb, what is the standard or the normal length of telomere and if it is on all cells of the body or it's specific?
Yeah, you have part of the answer with the former, with this one.
It's different.
You cannot generalize, you
No.
cannot say that, okay. But I think that in the future, as a kind of personalized, dedicated measurement, you will have the possibility to know not only what are the length of the telomere in your skin biopsy, whatever, but and interestingly, you will have the trajectory of the telomere length. And that's something that could be considered maybe as a good biomarker of aging. All the telomere are going to be shortened year after year.
With the difference. No, it's not the difference. No.
What?
With, no.
With the allelic difference?
No, it's not. It's not like
Yeah, but
it's
I'm not sure that this is something that other question. It was a very good one. Yeah, yeah.
It's basic one. Maybe you said it, and I didn't understand why they chose the fibroblasts.
Do you have an idea? The other one? Why they chose this cell? According to you?
because maybe we can put them easily.
Yeah, these are cells that are very easy to cultivate, to grow, to manipulate, etc. We can do everything and we can collect them very easily. But at the end, it's during the first question. I think that it should be very, very interesting. And I think that has been done because it's a natural genetic paper. I think that in another natural genetic paper, they try to identify exactly the same thing, but in different cell type. And finally, but some people also say that finally the telomere length of your fibroblast as well maybe as the leukocyte. That's why just before I was saying that it could be interesting in the future that maybe only with some blood microdata of blood should be possible to analyze the leukocyte telomere length, because finally it should be used as a biomarker. You are not going to collect some liver or some heart just to measure the telomere length. Okay, so that means that it could be interesting to get some accessible cells and consider them as a kind of global biomarker. But the good question is to know that.
But there is a lot of type of leukocytes.
Yeah, but even though it's not a problem, I mean that you have to change your mind about the fact that we are not guessing if this leukocyte is more important than this one. No, it's to consider leukocytes as a kind of signature of what's happened in your body. So you don't have access to many cells. I mean that in reality you have access to skin cells, but it's a little bit invasive. You can have access to some swab, you know what it is, when you collect the saliva, you have some cells, also the swab. You can have access to some blood cells, okay? And in these blood cells, and it should be very interesting to know if what you see in the blood cell could be something that's happened in your body. So for that, there are a lot of different studies, and unfortunately, we don't have time to speak about that today, but that try to compare what's happened in terms of telomere length and shortening between some blood cells and other cells that are maybe more important, of course, such as the heart and liver and whatever. Okay, so that's good, or even for endothelial cells. I have another question here.
Do you know if there is a big variation in between individuals on the length of telomeres? Is it shortening faster in some people, or is it more on a homogeneous process?
It's aléatoire.
Yeah, this is a very difficult. I mean that this is a very interesting question, and maybe part of the answer should be brought by the analysis of here. No, after or before, I don't know here. Okay.
Sorry.
You know what I mean? The mother and the two sons.
Yes.
You can see that even between the mother and the two sons, we are not equal. And of course, that could be, I mean, justified by the fact that this is non-Mendelian and the repartition is not something that could be equal. But the question is, I mean, your question is more interesting than just this kind of snapshot, because you ask a question about the rapidity to telomere loss. That's the key point, I guess. That's the most important thing to know if we are all equal in terms of, and even, I mean, that if you go further on this question, that would be very interesting to see if regarding different challenges, if we are all equal or not. If I expose to UV or to stress, whatever the stress we can consider, like metabolic stress or inflammatory stress, I would love to know if my telomere are responding the same than yours, for instance, and if we are equal. And if we are not equal, you have also to consider that you are just, I mean, that looking at a kind of result. Telomere length is just a result between telomere synthesis and telomere degradation.
They suggest that the key point is not the erosion of telomere, but the conservation of.
Some protections are not efficient. We just saw that with the TRF2 experiments. I mean, that if you are not protecting your DNA, so the capping stuff is very important, that means that you are going to lose more DNA. Okay? And also, after that, is not the end. I mean, that even if you lose some DNA material, we are not all equal on the DNA reparation processes. Okay? So, this is something that's fascinating, but this is something that is difficult to generalize. Other question, other remark? I have a lot of questions, but I don't want to spend too much time. Just one thing that, I'm in the lab, can talk loudly, but one question that I have is that if the finding of this allelic telomere length variability consistent across different cell types, you already had the question, or in diverse population, are they investigating it? So Mariam, this is something we just discussed, and I hope that the answer was good enough for you. Of course, if you have more questions about that, maybe we can try to find other publication. Well, try to retain important stuff, because this kind of publication is difficult for you. What is the taking home message here? It's quite simple. They're comparing different strategies. They are starting from, and thanks to Farah, we have a very clear explanation of what is the starting point. The starting point is the TRF and the Q-fish. According to me, if we can go back to that, you have to know what are the advantage and the limits of this kind of technique. Okay, for the exam, I think that it's quite easy for you to understand exactly what they did. Okay, okay. We are not going to discuss because we are not going to spend too much time. This is very interesting, but we have to go further, and you can see the different limits, etc. I have just maybe one question. Do you know if these two techniques are, and this is a really naive question, are still used in today? I mean that the TRF and the Q-fish technique are some techniques that are used in the different lab, or they are all using stellar technique. We don't know.
I think TRF is the most widely used.
The one that is more used in the world.
Yes, the reference.
But we are okay to say that this is not something that is used in routine clinically in the hospital. This is not used today? You don't know. Does anyone know? It should be interesting to know. Maybe for the next one, for the next workshop, I'll try to get the information. If some people are here in Toulouse, we are not using that. We would love to, because I'm in connection with the gerontopole. They would love to measure the telomere length. This is something that we could do. But of course, we are not the specialist of that. So that means that, first of all, you have to do the collection, to do the experiment, and to do the analysis. When you consider 1,000 patients, for instance, in the INSPIRE cohort, I think that could be difficult. But that's something if I remember, that is added to the protocol. So TRF, Qfish. Just one question here again. Love it because some stuff I didn't know. Telorect and tel time. I didn't know these words particularly, and I think that's just very interesting. Here, of course, you don't have to know that, okay? Because it's quite difficult. It's difficult to explain. It's difficult to understand. We are not molecular biologists, and it's not a problem. Okay? But I didn't know this finally this word, and you take it and you explain it very well. Okay, I didn't remember that it was a publication. What was the aim? Okay, what was the aim of the publication? I think that you know it. What was the aim of the publication?
Like in order to overcome the limitations of the other methods.
Yeah, of course, it should compare different techniques and to see that if we can propose a new one. Several dilution, telomeric specificity. Can you explain the validation? You talk, I think it was your when you talked about one moment about specificity, and I didn't understand exactly what was here.
Yeah, I'm not sure to explain.
I'm not sure
why.
also. It's not a problem.
I think it's because here they can.
To be sure that we are amplifying telomere, what we have to do to be sure that we are amplifying a telomere and not because that the main risk is what? To amplify some non-telomere DNA. So to be totally non-specific.
Yes.
So I think that they introduced this kind of primer, telomere specific one, but in the gel, I didn't understand exactly what was it. And you mentioned during the commentation.
Sometimes they don't put all the components of the analysis.
So the one that is interesting for us is the first one, XPYPE2. I say that for the people that are online. This is the one that is interesting for the reaction MABC. And for the other one, that is a negative control, saying that we are not.
For the chromosome
Yeah, we are not amplifying the non-telomere material. So that's something that is okay to say that we are specific from the telomere. Okay. Okay. Explant of fibroblasts, we already discussed about that. What is the investigative band? Oh, yes, and the last one. Other question, other remark? Yeah.
Quand on est, on n'a pas tous la même longueur de télomère, alors. Ok. Mais
when we born, we are not with the same level of telomere length.
Ma question, c'est peut-être bête, mais si on arrivait à savoir à cet âge, enfin à la naissance ou très tôt la longueur des télomères, est-ce que ça pourrait pas servir de biomarqueur pour précoce de peut-être développer des maladies à l'âge plus tard?
She hypothesized that maybe knowing the telomere length at birth should be very informative for the predictive score of developing some other pathologies in the future. This is an open discussion. Maybe you watch too much movies because this is something that is in the, I don't remember the name of the movie, but they are a couple, they are supposed to get a baby, and we say that this baby, what is the name of this? Welcome in Gadaga. And this is a good question. This is a dramatic but a good question. Are we only, dedicated to telomere length? That means that I think no. Yeah, my point of view, and I just say no. We are not only connected with telomere length. Telomere length is not the alpha and the omega of everything. What is your opinion? Should be very interesting, very informative. Do you do that if you have the possibility to measure in your future newborn children to measure the telomere length and to get a predictive effect? Say that, okay, at 25 is going to have a kind of cardiovascular disease, then at 42 is going to get something like start some cognitive defect, and at 65 it will die. No. So this is something that is philosophical question, and that's very important. And we are on this kind of question. You have to differentiate the philosophical question and the medical and biology question. That's a very passionate discussion. And the discussion of today is also to know if we can identify, and you will maybe do that in the future, is the biomarker of aging related to different pillars, such as inflammation, senescence, telomere. So I think that at the end of the discussion, we can consider telomere as a pretty good marker. If we consider the two first articles, that's very interesting. But this is difficult to do. A lot of control of different things can be affected by different things. So we have to take care about what we propose with telomere length. Okay? Was the question the last one, maybe because we have to switch to the last group? No, thank you again, David and Farah. You did a very good job. Okay, the last one. The last one. Oh, pourquoi?
On a fait la revue qui avait des sur le gun.
C'est pas grave, c'est légal, c'est mieux. Si tu as oublié de prendre, on va mettre une c'est pas ça. Ça va être bien parce que elles ont fait la revue. They did the review instead of the article because I realized, and it's my fault, so that there is a kind of match between what I sent you and you know it's difficult because I don't have all the email address. I have Kira in one side, I have the CCC in the other side. I have mine, of course, that I know, but it's difficult. You don't have access to Moodle. That's good to finish on that, finally. That's very good. I prefer to do that rather than the last paper. So the last paper was this one, a very short one, published in Nature, showing for the first time that there is a shortening of telomere in H fibroblasts. So it's quite easy to understand regarding the discussion we had with Farah and Apolline to say that there is a shortening of, but it was the first one, the most important one. That's why it was published in Nature, okay? So let's finish by a kind of global review of everything. Here, maybe we can interrupt them to just to try to be sure that at the end of the day, we will be on the same page regarding the use of telomere length as a good biomarker of aging. Le partage d'écran est en pause avant. Pourquoi? C'est bon. Can you see the screen only online? Give me a yes.
I can see Zoom. I don't see a presentation.
This is a beautiful picture of a chromosome generated by ChatGPT, I guess. No. That means that you have to pay something to someone here. Okay, do it again. No, you are. Look at you are sharing this screen. So go to No, just go up. Here, nouveau partage. Et là, tu choisis non, c'est de gauche. Elle est en plein écran. Ouais. Vas-y, partagez. Janine, do you have us? Yeah.
Yes, yeah.
That should work. Have you seen this beautiful chromosome? Okay, so we are finishing by a review, which is not too bad. And okay, let's maybe interrupt them if you have some question, or we can ask. I mean, let's open the discussion at the end. You can go.
Hello everyone. We are going to present this article called Telomere History and Hallmarks of Aging, published in Cell in 2021, which discusses the impact of telomeres and on aging and age-related disease.
vais essayer les cafés comme voilà.
First, as we know, global life expectancy is increasing around the world, and in 2050, there will be around 2 billion older people than 60. But there are more diseases like cardiovascular disease, neurodegenerative disease, and cancer. That's why it seems important to understand aging in order to understand its mechanism and mitigate its effect. With the main objective is to remain in a good health, called the health aging.
Telomeres represent the end of the chromosome, which are therefore the terminal parts of the DNA, and those purpose is to protect the DNA. Telomere comes from Greek, and telos means end, and meros means part. And they are made up of a repetition of nucleotide sequences, and they are protected by a complex of proteins called sheltering. This was discovered in 1913. It was shown that cells had a certain number of limited division, and finally, it was in 1980 that the existence of a protein called telomerase was demonstrated, which is capable to maintaining the length of the telomere, preventing the loss of these nucleotide sequences, and so the mechanism was established between telomerase and cell aging.
Surprisingly, the TERT mice exist. What does that mean? That means that the mice mutated for the TERT one. We have the fish also at lab here. If I remember, this guy are aging really super rapidly, but this mutation is not lethal. So you will find different paper with TERT and that mutation. And in my mind, I guess, if I remember, that is not lethal. So I think that there should be maybe a kind of a repetition of different things, but this is not lethal. And here in TR2, you can imagine that this is very important. And now compare, I mean, that regarding the first paper, you can see that if you don't have this TRF2, of course, all the other things are useless. Okay.
Because the aim of telomere is to maintain genome stability during cell replication, as we see in each cell division, telomere are shortening. But in some cells, like in stem cells, germinal cells, there are an increasing of activity of the telomerase to make length grow, to avoid senescence in the cell, in particular germinal cell and stem cell. As they exist some disease called telomeropathy, which is due to dysfunction, dysregulation of the maintenance of telomere due to a mutation of TERT or TERC, and cause some degenerative disease. So we can say that some people who have a mutation of TERC or TERC are shorter telomere than other people.
Even if we cannot to reply to, I don't know who asked the question, we cannot reply to the fact that we should have a kind of minimal length of the telomere. We don't know exactly if it's good to have, let's say, 7 kb or 10 kb, whatever. But what we know is that if they are too short, there can be some complication. And here, I don't know if you're going to switch to that, but some people that was in Master 1 with me know that some pathologies are unfortunately and rare diseases are due to this kind of alteration of this is not really the telomere. It's you know, the Werner syndrome. Remember these children that are aging very highly and very fast, and that because the helicase, which is an enzyme which is able to open the DNA for the telomere, okay, for the telomerase, and also the Huntington disease, of course. Okay, sorry for interrupting.
So we are talking shortly this article telomere shortened during aging of human fibroblasts. To resume, this article is published in 1980.
Yeah.
And briefly, it shows that the length of telomere is decreased with age. This fibroblast is fetal, and after with age corresponding in vivo age, and we see that during the evolution of the age, the length of telomere is decreasing. This part is subterminal part of the telomere because it's GGGG, and we all see that in the other fibroblast, the length is decreasing. And to resume, they make a statistical model, and they show that for older people, the telomeric DNA is decreasing, and the length of telomere decrease also. So this article showed in these ages that the telomere has play an important role in aging.
Just to be sure here, maybe to anticipate some question, if you can come back to the previous one again. It is not the same patient,
No.
right? It is not the same patient. We don't have fibroblasts from fetal state to the 91. That's a criticism. That's a criticism because if I understood the discussion we had before, we don't know exactly the capital we have when we are born, and that I think that it should be very interesting to analyze the loss or the progressive loss of the telomere length. Here we are just saying that, but we don't know if the individual at 71 did some exposure to whatever during all this lifespan, etc. So we don't know that. And so that means that it's quite difficult to make a kind of, even if this publication was a kind of very important one, because for the first time it has been shown that we can link the length of the telomere to the age of the donor. That was the proof of concept.
Recently, Lopez identified what were known that all marks of aging, biomarkers of aging, including the role of the telomere, which are all interconnected and involved. Telomere dysfunction influence various aging processes like cellular senescence. Short telomere trigger senescence, which affects tissue regeneration by the activation of the p53.
As we already say, in each division, telomere are shortening, and when they are too short, there are an activation of p53, which cause block proliferation and induce apoptosis to cellule and death, and so degeneration of tissue.
You have to know that that telomere shortening is a kind of protective system, finally. Because if we understood, after this loss of protective system, I start to lose DNA. In case of, and even before the loss of DNA, I'm activating a program that is dedicated to stop the cycle. So to not reproduce cells that will be probably affected by mutation. So this is a perfect physiologic adaptation to life, finally. But this system, unfortunately, is highly activated during aging. That's a normal situation. We are talking about normal aging, finally. That's a marker of normal aging.
It's also interconnected with stem cell depletion. Progressive telomere loss lead to a reduction in stem cell affecting tissue with a high regeneration rate. With genomic instability, telomere shortening induces chromosomal abnormality and favors the development of cancer. With mitochondrial dysfunction, the link between telomere and mitochondria highlights the importance of mitochondrial function in aging through the increased production of reactive oxygen species and oxidating agents. With epigenetic dysregulation and loss of proteolyte, telomere also affect epigenetic mechanism and protein stability, directly influencing neurodegenerative disease. And with impaired nutritional sensitivity, telomeres have an impact on the ability of cells to respond to metabolic signals. And there's the role with the inflammation. Telomere dysfunction trigger chronic inflammatory process, contributing to inflammatory disease and aging. In summary, during normal aging, somatic cells are designed to have a certain dividing cycle, with diminution after a certain time. When the telomere becomes too short, with a length that is too short for the cell to divide, the cell enters in senescence, and this reduction in telomere left impact on stem cells.
And after, in this article, they talk about the link between telomere and cancer. And we know that when there are an inactivation and p53, there are non-controlled proliferation, we will call cancer. Therefore, there are more cancer in elderly people and mainly in cutaneous cancer due to involving the epithelial cells.
This also open a therapeutic prospect, particularly for influencing aging. In this case of good aging, telomerase activator can be used. So it's clear that the aim will be to mimic the action of telomerase in order to extend telomere, and this increased cell lifespans. But in certain cases of cancer, the most interesting option would be to use telomerase inhibitor, which is already possible in clinical practice.
That's a very interesting point, because we know here the perspective, of course, of telomerase activator. I'm surprised that we don't have the question before, but maybe some potential activator of telomerase. What is the risk that could be raised by the use of this kind of molecules? Activators of telomerase?
Cancer?
Yes, of course. Which cells are using this kind of process very easily? These are the cancer cells. Or again, exactly the same with the senescent processes. Now, you know me, and you are used to hear that if you are considering senescence as a cause of the target, you have to think about what to do with this kind of senescent cell. If you try to reverse, and here again, if you try to activate the telomerase, you should be able also maybe to increase this kind of resistance to the cancer cells. And what about telomerase inhibitor for cancer cells? Yeah, so you can generate, but here, if you do that, you are going to increase the aging process, no? What do you think about it? You know that you already heard about the fact that chemotherapies are promoting the acceleration of aging. You know that there are some studies, and I already told you, for those that are in the UAG, maybe the other one never heard about that. But there are some studies showing that if you have some pediatrician cancer, so when you are aged between 6 and 15 years old, if I collect some fibroblasts when you are 40, 45, 50, I can see that the senescence is more increased than in the global population, meaning that, of course, the damages that have been done by some drugs in the past could reflect in the future and can precipitate the aging processes.
So to conclude, we can see that telomeres play a major role in aging, and in improving our knowledge of them would open up new therapeutic prospects. Telomerase restoration therapy is being discussed as a potential approach for treating degenerative disease. However, the risks need to be better understood because the link between telomerase activator and cancer. So we see that it's important to understand mechanism of aging to understand better the mechanism and to develop some treatments to improve life and avoid some age-related disease.
Okay, thank you very much. We did two different publications. We did the one that you are supposed to do, and finally you did that in one slide, and I think it was enough because the message is a good one. So question is, after that, can we consider telomere length as a biomarker of aging? What do you think about that? What is a biomarker? Biomarker should be a little bit specific, but what do you think? Maybe you, maybe you did the review, you think that telomere length can be considered as a biomarker of aging, or just only, this is what you propose, a kind of way of action to fight against aging?
No, it's a real biomarker of aging because in normal cells without disease, the telomere are shortening, but
that's a physiological state.
Physiological state, but we see that it's interconnected with all over the biomarkers, and it's difficult to see which being the first, and it's a certain.
Well, this is a question we will have maybe at the end of the week, not of the week, because we're going to see after in November. But that's a huge question to know. I mean, what is the triggering point? What is the first things to happen? Finally, maybe we don't care about what starts firstly. We don't want to know exactly if I don't know. I mean, the cause of senescence can be multiple, such as oxidative stress, such as DNA damages, and this DNA damage can be done by oxidative stress, which can be also as a result of what's happened during when you are young, etc., etc. But at the end, here again, we are talking about some biomarkers. So we would like to know if we can maybe connect some marker. Here today we are talking about telomere length to maybe age-related pathologies or a state of aging. I don't know if you can doctor that. Maybe it's a frailty or something like that. A question today regarding the different publication. What do you think about that? Did you hear about specificity of pathology? Did you hear about a difference between pathologic and physiologic? What do you think about that? We know that when we age, we are losing some telomere. The first publication demonstrates that if the telomere, the telomere process construction is altered, we are going to have an acceleration of aging, particularly in endothelial cells and in the cells. But we don't have very important evidences showing that because you are losing your telomere in the liver, you are going to have hepatotoxicity or whatever. This is something that is intuitive, of course. But the first question is why I should have some shortening of my telomere in the liver specifically. Because I drink too much, because so I am affecting some part. That could be interesting, I think, in the future. If we want to really think that telomere shortening is specific of age-related pathology, and such as cardiovascular disease, Alzheimer's disease, whatever, we measure the telomere length in different contexts. And to say that, okay, in a population of Alzheimer's disease, we can see that rather than accumulation of amyloid plaque, we have also a shortening of the telomere. The problem is what? The problem is still the same. We are working on humans, so that means that we have only cross-sectional study. We don't have a longitudinal one. You are not going to ask your patients to collect some neurons from the age of 20 and every 10 years. So that means that when they are died from Alzheimer's disease, you are going to collect the brain and to have some possibility to measure the telomere length on the neurons. And you're going to have to say, okay, this guy is, of course, has some telomere that are shorter than the other one. Is it the cause or is it a consequence? We don't know. We don't know. So that's why the mouse model and the model, the first one, is very interesting because we are trying to induce the loss of telomere specifically in different tissues and to analyze. Some studies clearly demonstrate that if you are targeting neurons, particularly by telomere shortening, you are going to induce some neurodegenerative process. Not Alzheimer, not Parkinson, but some neurodegenerative disease at the global point of view. Okay, so that's interesting. It's also the same if you are targeting specifically. We're going to see that we saw that in the first publication. specifically, you can also affect the vascular disease, etc. Okay. So to be a good biomarker, it is something that could be also a kind of a predicting model. We didn't speak about prediction here today. Why? Here again, because we are considering telomere length as a kind of situation at one time point. Okay. It should be really informative to get maybe, and maybe in the future, I think that this is something that will be done. The use of telomere length should be something that could be measured longitudinally, and to try to see if the slope of the cliff, because all of you in the room will have telomere length, shorter in 1 year than today. But the question is not here, is to understand first the consequences, and second the slope of this loss. If you are losing something like, I don't know, 40% in 1 year, you are losing a lot, too much. And here we can talk about the strategy that are here to slow down this property. I don't know if some strategy are really working. I'm not an expert on that. If you're interested by that, it's something that you can double check. But at the end, I think that using telomere length as a good biomarker is difficult. Okay, it's quite difficult because of the cell specificity, because of the kind of personalized shortening, because of the cross-sectional stuff regarding the longitudinal and follow-up, etc. But you understood today that this is something that we are measuring, not in a routine, but for biology study. This is you should know how we are measuring this kind of process. You should know how, not in details. I don't ask you to be a specialist like a para-hematopoietic. No, but I would like to say, okay, we are measuring by, we know what it is, by PCR, etc., etc. So you should know exactly how we are measuring. You should know the techniques. We talked about TRF, QFISH, and Stellar. So maybe this is something that you should know. You should also know what have been discussed by Estelle and Aurore and Estelle and Aurore. You should know exactly. First of all, how can we link the telomere shortening and aging, just aging, not age-related pathology, but just aging. The paper published in 1980, and then they found the review. I don't know how did you do that, because I didn't find on the model, so I guess I removed that. But if it's not the case, what I will do is that I will put again the review. It's quite interesting for you to read that. Okay, in the review they are just explaining what they just Aurore and Estelle presented, different things about what is telomere, or it is used, as a very easy to understand level. Okay, it's not too complicated. Do you have any question? Any suggestion? What about the presentation? It's difficult to discuss about the review. Do you have maybe precise question about the paper? Nature, Donald? No? Nothing else? Do you have question about the today topic? Yeah.
Can you do a longitudinal study on mice? Like,
I think it's more easy to do that in human than in mice. In mice, it's quite difficult, for instance, if you want to work on, I don't know, skin or whatever, you are going to make some biopsy. So it's difficult, and to try to see in a different part to do this biopsy. Of course, if you want to collect part of the brain, you cannot. By definition, longitudinal study can be done only on non-invasive collection. That's why I was talking about leukocyte, etc. So this part is a part that comes only from my brain. So that means that at the exam, you don't have this kind of thing. But I would like you to imagine the biomarker of tomorrow. And by this way, this biomarker could be non-invasive, predictive, easy to do, and with how can I say that? With a high power to anticipate, or at least to characterize the pathology associated with age and the loss of function. Okay. Any question, suggestion of today? Are you ready for tomorrow and for next? No, not tomorrow. For Wednesday and Thursday. What are the different workshops we have? I don't remember in the good order. What is the Wednesday one? Remember? Who is dedicated to Wednesday? What? Go ahead. Yeah, you should. Do you want me to present, to show you the table to be sure that everything is covered? Or it's okay for you? So you are going to present on Wednesday? Yeah, one group, you are free. What is the topic? Metabolism, inflammation, or senescence? I don't remember. Inflammation into?
No, inflammation and
The last one, on November 14. So senescence or metabolism, or oxidative stress or mitochondria? Okay, mitochondria. So one group and the other one. Online, you're well, two online. Okay, why not? But I have your name, so your name is on the table. Okay, or maybe you plan to do that tomorrow. If you will do that tomorrow, please. So yeah, you know the same thing. Please now you know exactly what is the game. You know how it works. I want you to get the different information from the publication. It's quite good what has been done today because the people that present today try to make some also to put some background, some explanation. You know, don't be just only on the publication. You can also try to understand exactly what you are saying and what you are discussing. Even if you don't have too much time, and this is something that is important to do that in a shorter time. We are talking about shortening telomere. You don't have. Sometimes you won't have very long time to prepare your presentation in the future when you'll be researcher. So that's a good training also for you. Yes.
I think someone already sent the mail, the 14th of November.
Yeah.
The BSG, we have another course.
Oh, really? Again? Because I changed that for Cecile's students. Because they should. And in DAG, normally you have nothing.
Si.
Ready? Vous avez quoi? Après, hein? Ah, mais c'est. Oh. Je suis de Camille, alors c'est ça. Alors, on ne peut pas le faire deux fois, moi j'en ai marre. Normally, I do that in the same week. But because of Cecile students, we have to change.
Ouais, présentation juste.
On verra bien. Mais normalement, moi j'avais tout vu avec Héloïse pour que le temps soit bon. Vous regarderez ça, vous n'inquiétez pas. Bon, mais pour mercredi, en tout cas, vous êtes OK? Vous revenez avec des questions? C'était pas mal, hein? C'était pas mal, hein? Par rapport à d'autres années où c'était pire, c'était pas mal. Mais il faut encore plus que vous voulez. Any extra things to discuss? If you don't, thank you very much and see you on Wednesday at the same time. Merci beaucoup. Okay, you will have questions. Yeah, you know, today, you know, say we discussed about rise and forms. Not only Est-ce qu'il vous l'a ouvert à ça? Ah, mais c'est bon, tu l'as ouvert. Alors, on va pas compter. Après, je vais la dame du ménage, je pourrais qu'elle soit la dame du moment, c'est la dame. Avec elle, on arrive à. Elle est arrivée dans l'entrée, mais par contre, c'est la boutique, c'est que dedans, quand même, vous travaillez. Ne volez pas les dents. Là, il ne s'occupe pas de voler les dents. C'est plus d'avoir. Il est sûr. Les banques, dans quel